Okay, so let's start the pre-test version A of biology, which is given here. Yes, so this is the pre-test document link. Just a minute, it's redirecting. Okay. Okay, so this test should be done within 20 minutes. So I will set Google timer here. So this will be 20 minutes. Okay, so I will start this. So here the question. So this is a structure of cell, and using this diagram I have to answer the following 6 questions. So these are A, B, C, D, E, F, G, H, I, J, K, L. So the first question is levels G represents several proteins that assist with cell division, protection, and maintenance of cell safe. So what do these proteins comprise? Level G means level G is here. Okay, and the options are cell body, cytoskeleton, head, musculoskeletal system. So G is here. So actually I have little knowledge in biology, and for a long time I have not in touch with the biology subject. So I will guess it. I think it will be it is not cell body, maybe cytoskeleton, not a head on musculoskeletal system, maybe cytoskeleton or musculoskeletal system, but I'll guess it. I think it will be cytoskeleton. Okay, so next question is that level F represents what? Centrioles or microtubules? Level F. F means this. Okay, level F represents this which is necessary for test. So centrioles or microtubules? So I will go for centrioles for cell division. So next question is that the organelle represented by level D. Level D means level D, and is mainly found in which portion and what? Your answers should be the names of the organelles. Separate your answers with comma and then a space. Okay, so level D is here, so there is some rough part and also some smooth parts are there. So I think it will be rough endoplasmic reticulum, and it remains in cytoplasm. The main function of Golgi apparatus represented by level which, either L or K. Golgi apparatus, L or K. Yes, this is L and this is K. K is the correct answer, I think, because this is the Golgi body. So K for lipid synthesis or K sending receiving proteins. Golgi body actually transfers the proteins, so it will be sending and receiving modified protein. Next is that the organelle level C is present in C is here, so the organelle present in that part present in animal cells, plant cells, both animal plant cell and prokaryotes. So I'll go for animal and plant cells. Next question, the organelle labeled A or C or E or H will be in higher numbers in muscle cells compared to fat cells. So A, C, E, H. A, C, H and E. E means that part. Okay. So I think H is mitochondria. So H, so which requires in muscle cells. So I'll go for H. Move to the next part. Okay. Just as an amino acid is a monomer of a protein, a dash is a monomer of DNA, pentose, nucleotide, nucleoside, phospholipid. So DNA nucleotide is the monomer of DNA. So nucleotide will be the correct answer. Now a DNA monomer is called a nucleotide and is made of three parts: nitrogen base, deoxyribose sugar, and sulfate group. Which part is incorrect? Oh, this. So it should be a phosphate group. In place of phosphate, it is written as sulfate. Okay, so this is incorrect. So sulfate will be the correct answer. Sulfate group. DNA is more or less stable than RNA and therefore less or more reactive. So I think DNA is more stable than RNA because it has double-stranded structure, whereas in case of RNA it has single-stranded structure, so it will be less stable than DNA. And DNA is less reactive than RNA because it due to its double helix structure, double-stranded structure, it is very difficult to compact or to form the complex structure, whereas in case of RNA it is not so hard to form the complex structure. So I can justify it to say that DNA contains hydrogen atom, whereas at 2 prime end atom at 2 prime end, whereas in RNA it is which group? Okay, this is the justification according to me. So move to the next question. Use this diagram to answer the following 7 questions. So there is a delta U C G C G A epsilon. So here U means uracil, C means cytosine, G for guanine, and A for adenine. So according to the strandedness of this molecule, what type of nucleic acid is? So I think as there is adenine and it should be RNA, DNTP, NTP or DNA. So it should be DNA. Next question, according to the types of bases. So if there is a uracil, so that should be RNA. According to the types of bases in the molecule, what type of nucleic acid is it? DNA, RNA, DNTP or NTP. So it should be, I think, RNA. Now, what is the paradoxical about this image? So, DNA contains A and T bases, whereas and double stranded structure, whereas in RNA has single stranded structure and AU basis, that is adenine and uracil. So according to your answer question 11, what bases do delta and epsilon represent? Adenine thymine, adenine uracil, thymine adenine, uracil adenine. 11 means that is in DNA, so DNA remains adenine and thymine. So it should be adenine and thymine. According to your answer to question 12, what bases do delta and epsilon represent? Adenine, thymine, adenine, uracil, thymine, adenine, uracil, adenine. In question 12, 12 means the answer is RNA. So in RNA that remains adenine and uracil. Okay. Use the following diagram along with the previous to answer the following two questions. Sorry. So this is a sugar molecule from the previously discussed nucleic acid, and there is a hydroxyl bonded to the 2 prime, 3 prime and 5 prime carbon. What type of nucleic acid is it? So in 2 prime we know that if it is a hydrogen then it will be deoxy ribonucleic acid, but in case of there is a OH group in at 2 prime end, so it must be oxyribonucleic acid. So RNA, that is ribonucleic acid. This is the correct answer. What conclusion can be drawn from your finding? DNA can sometimes include uracil. The backbone of DNA can sometimes include this type of sugar. The amount of hydroxyl groups does not matter. RNA can sometimes be found as double-stranded. So according to me, this should be the backbone of DNA can sometimes include this structure. So next, the ordinary flow of information in a cell is dash dash dash, separate each answer with a comma. Flow of information. So actually it comes from DNA, then it moves to RNA and then protein. So it should be written as DNA, then RNA, then protein. Okay. So the process that takes place at the first arrow is what, and the process at the second arrow is what? Okay, so RNA synthesis, protein synthesis, next ribonucleogenesis, proteogenesis, transcription, translation, DNA cleavage, codon anticodon interaction. So I think it will be DNA cleavage and codon anticodon interaction. Which statement about proteins is correct? An alpha helix is indicative of primary structure. Beta sheet is indicative of secondary structure. Multiple proteins interacting is indicative of tertiary structure. Quaternary structure is only theoretically possible. So I think alpha helix is not a primary structure, it is a secondary structure. Beta sheet is indicative of secondary. Yes, beta sheet is secondary structure. Multiple proteins interacting is indicated. No. Quaternary structure is possible. So I'll go for beta sheet. This is the correct answer. Okay, so there is some amino acid. Use this diagram to answer the following three questions. A DNA template strand contains this sequence 5' GAG, AAC, TAT, AGT, CTG, 3'. So what is the complementary strand of mRNA? So in place of 5' it will be 3'. In place of G it will be C, then A U G C. So C U C, then Q U G, then A U A, then U C A, then G A C. Okay, so 3 B will be the correct answer. Now your mRNA strand of choice must be reversed before converting to amino acid. So it must be written as DNA again, complementarily to the answer to the previous question as tRNA from T 5' to 3'. So as it is reversed, so it will be 5' to 3'. What protein sequence does this mRNA code for? Notate each protein as its 3-letter abbreviation and separate each protein with a hyphen. Okay, so first part is CUC. So CUC will be leucine. Then UUG. UUG, it remains in leucine. Leucine. Then it should be serine. Then UCA. So UCA again. Arginine. Next, asparagine. Okay, I also move to the next part. So a point mutation causes an amino acid to change to a stop codon. So what is the effect on the resulting polypeptide? No change, truncated and non-functional, doubly effective, goes to nucleus to function. So goes to nucleus to function. Second point mutation causes that stop codon to change into a codon that is different from the original. So what is this mutation called? Silent, nonsense, missense, intel. So I think it will be a second point mutation causes that stop codon to change into a codon that is different from the original. Okay, missense. So okay, I have completed the test of biology A. Now I will check the score. So 17 out of 25. Okay, so this is correct, and okay, this also correct. This actually correct answer because RER means rough endoplasmic reticulum and it remains in cytoplasm. So this should be the correct answer. Next is correct. organelle labeled C, that is also correct, and H, that is also correct. Sorry, amino acid is a monomer protein, that is also correct. DNA monomer is called nucleotide, that is also correct. DNA is stable RNA, that is also correct, more stable than it is. So what is the justification? The RNA has one more hydroxyl group, and which one more hydroxyl group than DNA, so it makes it less stable and more reactive or something similar. So these are close to my answer. This can be the correct answer. This is also correct. Now this is also correct. Now this is the paradoxical about this image. So double stranded is indicative DNA while the presence of uracil indicates RNA. This also can be correct answer. This is not incorrect. Okay, this is also correct. This also sugar molecule, and this is also correct answer. Congenitron. So this correct answer is that RNA can sometimes be found as double stranded. Okay. So this is the correct answer. Why it is taken as incorrect? Okay, that should be transcription and translation. Okay, so I will stop here for the pretest part as the timer completes. Now I will move to the second part, that is short break for 5 minutes and then start the self study. So in self study I have to copy paste each question I got wrong into Google and append AI, that is without AI knowledge, and then attempt to grasp the key concept of the question. Okay, so I will set first 30 minutes now. Yes. Okay, so I will start my 30 minutes. Yes, started. So the first wrong answer is that okay, this is correct, this is also correct, this is also actually correct, because rough endoplasmic and smooth, this will be rough endoplasmic reticulum in cytoplasm. So we can check what is RER. So what is RER in organelles, so I can study it. Okay, so this is the endoplasmic reticulum, and so the endoplasmic reticulum is a part of transportation system of the eukaryotic cell and has many other important functions such as protein folding. The word endoplasmic means within the cytoplasm, so it remains in cytoplasm, and reticulum is Latin for little net. So it is type of organelle made of two subunit, rough endoplasmic reticulum and smooth endoplasmic reticulum. The endoplasmic reticulum is found in most eukaryotic cells and forms an interconnected network of flattened membrane-enclosed SSCs known as cisternae, that is in RER, and tubular structure in SER, smooth endoplasmic reticulum. The membrane of the ER are continuous with the other nuclear membrane. The endoplasmic reticulum is not found in red blood cell or spermatozoa. So there are two type of endoplasmic reticulum that share many of the same proteins and engage in certain common activities such as the synthesis of certain lipids and cholesterol. Different types of cells contain different ratios of the two types of endoplasmic reticulum depending on the activities of the cell. So RER is found mainly toward the nucleus of the cell, and ACR, so this is the important point. RER is found mainly towards the nucleus of the cell and ACR towards the cell membrane or plasma membrane of the cell. The outer face of the RER is studded with ribosome that are sites of protein synthesis. The RER is especially prominent in cells such as hepatocytes. The ACL lacks ribosome and functions in lipid synthesis but not metabolism, the production of steroid hormones and detoxification. The ACR is especially abundant in mammalian liver and gonad cell. The ER was observed by light microscopy by Charles Garnier in 1897, who coined the term ergastoplasm. The lacy membranes of the endoplasmic reticulum were first seen by electron microscopy by Potter, Albert Claude, and Fulham. Sorry. So this is actually rough endoplasmic reticulum, and this is the smooth part. Okay. So I will move to the next wrong answered question. Mm, that is justification. Okay, so RNA has one more hydrogen than DNA, which makes it less stable or more reactive. Okay, why? I can say that why it is more reactive. So why RNA is which makes RNA less stable and more reactive. So comparison of reactivity and stability of RNA and DNA. So DNA is more stable but less reactive than that of the RNA due to the following reasons. RNA is single-stranded while DNA is double-stranded. RNA contains the deoxyribose sugar while DNA contains the deoxyribose RNA contains the ribose sugar and DNA contains the deoxyribose sugar, which means that DNA lacks one oxygen containing hydroxyl group. So the presence of an extra OH group makes RNA more reactive and less stable. That is the important point. So the nitrogenous bases of both DNA and RNA are hydrophobic, means water repelling, while the backbone of both DNA and RNA is hydrophilic. As RNA is single stranded, its nitrogenous bases are not away from the water. That makes it more reactive than that of DNA. So it is clear from the above discussion that RNA is more reactive and less stable as compared to DNA. Okay. So good point. Next wrong answered question is paradoxical. So it says that DNA double-stranded is this is also almost correct answer. Double-strandedness is indicative of DNA, while the presence of uracil indicates RNA. So I can write what is the basic difference. Basic difference of RNA and DNA. I can say the basic difference. The basic difference between DNA and RNA. Okay. So the most important point is that DNA remains the basis thymine, cytosine, adenine, guanine, whereas in RNA it remains uracil, cytosine, adenine, and guanine. Yes, this is the comparison chart between DNA and RNA. So DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. Function of DNA is that to replicate and stores genetic information. It is a blueprint for all genetic information content within an organism. And the function of RNA is to convert the genetic information content within DNA to a format used to build proteins and then moves it to ribosomal protein factories. And the structure of DNA consists of two strands arranged in a double helix. These strands are made up of subunits called nucleotides. Each nucleotide contains a phosphate, a 5-carbon sugar molecule, and a nitrogenous base. And whereas the structure of RNA contains only one strand, but like DNA is made up of nucleotides. RNA strands are shorter than DNA strands. RNA sometimes forms secondary double helix structure, but only intermittently. And the length of DNA is much longer polymer than RNA. A chromosome, for example, is a single long DNA molecule which would be several centimeters in length when unraveled. And RNA molecules are variable in length but much shorter than long DNA polymers. A large RNA molecule might only be a few thousand base pairs long. And the sugar in DNA is deoxyribose, which contains one less hydroxyl group than RNA's ribose. And the RNA contains ribose sugar molecules without the hydroxyl modifications of deoxyribose. And the bases in DNA are adenine, thymine, guanine, and cytosine. In case of RNA shares adenine, guanine, and cytosine with DNA but contains uracil rather than thymine. So base pairs in DNA are adenine, thymine, cytosine, guanine. In RNA, adenine pairs with uracil whereas cytosine with guanine. Location: DNA is found in the nucleus with a small amount of DNA also present in mitochondria. RNA forms in the nucleolus and then moves to specialized regions of the cytoplasm depending on the type of RNA formed. Reactivity of DNA is due to its deoxyribose sugar, which contains one less oxygen containing hydroxyl group. DNA is more stable molecule than RNA, which is useful for a molecule which has the task of keeping genetic information safe. Sorry. And RNA's reactivity is that containing a ribose sugar is more reactive than DNA and is not stable in alkaline conditions. RNA's larger helical groups mean it is more easily subject to attack by enzymes. DNA is vulnerable to damage by UV light, and RNA is more resistant to damage from UV light than DNA. So these are the basic differences between DNA and RNA. So next wrong answered question is that what conclusion can be drawn from your finding? So this should be the RNA can sometimes be found as double-stranded. Double-stranded RNA. Okay. So double-stranded RNA is RNA with two complementary strands. This picture is given here, found in cells. It is similar to DNA but with the replacement of thymine by uracil and adding one more oxygen atom. Despite the structural similarities, much less is known about tRNA, double-stranded RNA. They form the genetic material of some viruses, double-stranded RNA viruses such as viral RNA or siRNA can trigger RNA interference in eukaryotes as well as interferon response in vertebrates. In eukaryotes, the double-stranded double-stranded RNA plays a role in the activation of the innate immune system against viral infections. So Watson and Crick had noted early on that 2' hydroxyl group on each RNA nucleotide would prevent RNA from forming a double helix similar to the one they had described for DNA. Okay, so this can be possible, that double-stranded RNA. Okay. So next okay, this can be the backbone of DNA can sometimes include this type of sugar. Backbone of DNA can sometimes include that sugar, that is ribose sugar, include ribose sugar, ribose sugar. Okay. So DNA is basically polymer composed of two polynucleotide chains that coil around each other to form double helix. The polymer carries genetic instruction for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA and ribonucleic acid are nucleic acid alongside proteins, lipids, and complex carbohydrates. Nucleic acids are one of the four major types of macromolecules that are essential for all known form of life. The two DNA strands are known as polynucleotides as they are composed of simpler monomeric units called nucleotide. Each nucleotide is composed of one of four nitrogen-containing nucleobases, that is cytosine, guanine, adenine, and thymine. A sugar called deoxyribose and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds known as the phosphodiester linkage between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone, and the nitrogenous bases of the two separate polynucleotide strands are bound together according to base pairing rules A with T and C with G with hydrogen bonds to make double-stranded DNA. The complementary nitrogenous bases are divided into two groups, the single-ringed uh, pyrimidines and the double-ringed purines. In DNA, the pyrimidines are thymine and cytosine, the purines are adenine and guanine. Both strands of double-stranded DNA store the same biological information. This information is replicated when the two strands separate. A large part of DNA is non-coding, meaning that these sections do not serve as patterns for protein sequences. The two strands of DNA run in opposite directions to each other and are thus antiparallel. Attached to each sugar is one of four types of nucleobases. So it is the sequence of these four nucleobases along the backbone that encodes genetic information. RNA strands are created using DNA strand as a template in a process called transcription, where DNA bases are exchanged for their corresponding bases, except in the case of thymine, for which RNA substitutes uracil. Under the genetic code, these RNA strands specify the sequence of amino acids within proteins in a process called translation. Okay. So within eukaryotic cell, DNA is organized into long structure called chromosome. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, providing a complete set of chromosomes for each daughter cell. Eukaryotic organisms, animals, plants, fungi, protists store most of their DNA inside the cell nucleus as nuclear DNA and some in the mitochondria as mitochondrial DNA or in chloroplast as chloroplast DNA. In contrast, prokaryotes store their DNA only in the cytoplasm in circular chromosome. Within eukaryotic chromosomes, chromatin proteins such as histones compact and organize DNA. These compacting structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. So this gives long idea. Okay, so this is transcription and translation. So this actually given here, so I can find again. So what is transcription and transcription and translation difference? So differentiate between transcription and translation. Transcription, it is the first step in gene expression, and translation, it is the second step in gene expression. Transcription synthesizes RNA copies from DNA. Translation synthesizes protein from RNA copies. Transcription takes place in the nucleus of the cell. Translation takes place in the cytoplasm of the cell. Transcription uses RNA polymerase enzyme to make copies of RNA from DNA. The translation uses ribosome to synthesize protein from RNA transcript. It produces functional RNA and it produces protein. Here the only mRNA is involved, and here mRNA, tRNA, rRNA are involved. Transcription can be inhibited by antibiotic rifampicin. Translation can be inhibited by the antibiotic tetracycline. Okay. So next wrong answered question is that, okay, what is the sequence of mRNA code? Protein sequence. So actually mRNA means messenger RNA. So the role of messenger RNA is that in molecular biology, messenger RNA is a single-stranded molecule of RNA that corresponds to genetic sequence of a gene and is read by a ribosome in the process of synthesizing a protein. mRNA is created during the process of transcription where an enzyme RNA polymerase converts the gene into primary transcript mRNA. This pre-mRNA usually still contains introns, regions that will not go on to code for final amino acid sequence. These are removed in the process of RNA splicing and leaving only exons, regions that will encode the protein. This exon sequence constitute mature mRNA. Mature mRNA is then read by the ribosome, and the ribosome creates the protein utilizing amino acids carried by tRNA. This process is known as translation. All of these processes, form part of the central dogma of molecular biology, which describes the flow of genetic information in a biological system. As in DNA, genetic information in mRNA is contained in the sequence of nucleotides, which are arranged into codons consisting of 3 ribonucleotides each. Each codon codes for a specific amino acid, except the stop codons, which terminate protein synthesis. The translation of codons into amino acids requires two other type of RNA, transfer RNA, which recognizes the codon and provides the corresponding amino acid, and ribosomal RNA, the central component of the ribosome's protein manufacturing machinery. Okay, so next part is that what is truncated and non-functional. Point mutation causes an amino acid to change to stop codon. What is the effect on the resulting polypeptide? That should be truncated and non-functional. What is truncated and non-functional? Nonsense mutation in genetics, nonsense mutation is a point mutation. So that results in a nonsense codon or a premature stop codon in the transcribed mRNA and leads to truncated, incomplete, or possibly non-functional protein product. Nonsense mutations are not always harmful. The functional effect of a nonsense mutation depends on many aspects such as location of the stop codon within the coding DNA. For example, the effect of a nonsense mutation depends on the proximity of the nonsense mutation to the original stop codon and the degree to which functional subdomains of the protein are affected. As nonsense mutation leads to premature termination of polypeptide chains, they are also called chain termination mutation. Missense mutations differ from nonsense mutations since they are point mutations that exhibit a single nucleotide change to cause substitution of a different amino acid. A nonsense mutation also differs from non-stop mutation, which is point mutation that removes stop codon. About 10% of patients facing genetic disease have involvement with nonsense mutations. Okay, great. So next, okay, so all are completed. I will move to the next part. So next part is that next part is another 30 minutes. Then search this Google and append with AI. So this is the first. What is animal cell diagram? Okay, so I will start my Google timer again with 30 minutes. So again, the first question is that what is animal cell diagram? So this is the animal cell diagram. So it has ribosomes, cell wall, cytoplasm, nucleus, nucleolus, endoplasmic reticulum, both smooth and rough, centrosome, Golgi body, lysosome, mitochondria, microtubules, cell membrane, cytoplasm. Okay, so this is actually animal cell diagram. Okay, so now what is plant cell diagram? I think next question is that plant cell diagram. Yes, plant cell diagram. So this is the plant cell diagram. So in plant cell there is also rough endoplasmic reticulum, nucleus, mitochondria, nucleolus, Golgi apparatus, Golgi vessel, chloroplast, vacuole, ribosome, cell membrane, there is mitochondria, cell wall, Drax crystal, then rapid crystal, then cytoplasm, amyloplast, peroxisome, smooth and rough endoplasmic reticulum. So what is actually the basic difference between this? What is the basic difference between plant and animal cell? Difference animal and plant cell diagram. So, mean plant cell is surrounded by a rigid cell wall. Yes, that is the correct one. In animal cell does not have a cell wall. Okay, in animal cell there is no cell wall. Mm, presence of large vacuole is seen in plant cell, whereas in there are very small vacuole as compared to plant cell are seen in animal cells. These plant cells are larger in size, whereas animal cells are smaller in size. Plant cells have plastids, and animal cells do not have plastid. Centrosomes are absent in plant cell. Animal cells have centrosome. Plant cells do not have cilia. Animal cells have cilia. Lysosomes are very rare in plant cell. Animal cells have lysosome. So this is the plant cell, and this is the animal cell. Okay. So next, function of cell organelles. So these are the functions of sorry organelles. For example, nucleus. So nucleus remains in eukaryotic cell and it stores the genetic information. Nucleolus, it remains in eukaryotic cell. It makes ribosomes. In the cytoplasm, it remains all cells and contains the content of the cell. Cytosol all remains in all cells and gel-like matrix that holds water and nutrient. Cytoskeleton, it remains in eukaryotic cell. It functions as structure, support and transport. Ribosome, it remains in all cells and it functions to make protein. Rough endoplasmic reticulum. So it remains in eukaryotic cell, makes protein for endomembrane system. Smooth endoplasmic reticulum in eukaryotic cell and it detoxifies the cell makes lipids. Golgi apparatus remains in eukaryotic cell and it sorts and shapes protein. Mitochondria, it remains in eukaryotic cell, makes energy. Lysosome, it remains in eukaryotic cell, animal cells only, removes unwanted material and waste. Peroxisome remains in eukaryotic cell, regulate biochemical pathways that involve oxidation. Vacuoles remains in eukaryotic cell, store water and nutrients. Vesicles in eukaryotic cell transport materials around the cell. Cell membrane remains all cell, a thin flexible barrier that separates the cell from its environment. Cell wall, it remains in plants, fungi and prokaryotes, and rigid barrier that protects the cell. Large central vacuole remains in plants only, stores water and regulates target pressure. Chloroplast, plants only, makes food using the process of photosynthesis. So these are the functions of organelles. Now next RNA DNA. So this actually uh, shown in the previous part. Now it is deoxyribose sugar and ribose sugar. So the key differences between ribose and deoxyribose sugar is that the IUC name deoxyribose 2-deoxydiribose and it is 2s 4s 5r5 hydroxymethyl oxolone 2,3,4 triol. Structure hydrogen at carbon 2, hydroxyl at carbon 2. Also known as 2-deoxy-D-erythropentose and it is D-ribose. Discovered by Levin, Phobos Levin, and it was discovered by Emil Fischer and Oscar Piloty. Found in DNA, found in RNA. So this is the key difference, and the main point is that both sugars have an aldehyde group in their open chain form and serve as essential components of nucleic acids. So in DNA, it stores the genetic instruction, passes heredity information to the next generation, and ribose, it translates DNA's genetic code into proteins, acts in various regulatory and catalytic roles. Okay. The next is that central dogma of biology. So the central dogma molecular biology deals with the flow of genetic information within a biological system. It is often stated as DNA makes RNA and RNA makes protein. Although this is not its original meaning, it was first stated by Crick in 1957 and then published in 1958. The central dogma this states that once information has passed into protein, it cannot get out again. In more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is impossible. So information here means the precise determination of sequence either of bases in the nucleic acid or of amino acid residues in the protein. So he restated it in the Nature paper published in 1970. The central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information. It states that such information cannot be transferred back from protein or nucleic acid. A second version of the central dogma is popular but incorrect. This is the simplistic DNA to RNA to protein pathway published by James Watson in the first edition of the Molecular Biology of the Gene. Watson's version differs from Crick's because Watson describes a two-step DNA to RNA then RNA to protein process as the central dogma, while the dogma as originally stated by Crick remains valid today. Watson's version does not. So this is the basic difference of Watson and Crick model. Move to next question, that is DNA mutation type. Types of mutation. There are many different ways that DNA can be changed, resulting in different types of mutation. Here is quick summary. A substitution is a mutation that exchanges one base for another, such as A to G. This could be a substitution could change a codon to one that encodes a different amino acid and causes small change in their protein produced. For example, sickle cell anemia is caused by substitution in the beta hemoglobin gene, which alters single amino acid in the protein produced. Change a codon to one that encodes the same amino acid and causes no change in the protein produced. These are called silent mutations. Change an amino acid codon to a single stop codon and cause an incomplete protein. This can have serious effects since the incomplete protein probably would not function. Insertion. Insertion a mutation in which extra base pair are inserted into a nucleus in the DNA. Deletion means are mutations in which section of DNA is lost or deleted. Frame shift. Since protein coding DNA is divided into codons, 3 bases long. Insertion deletions can alter a gene so that its message is no longer correctly passed. These changes are called frame shift. Okay, so I'll move to the next part, that is uh, answer this question. So what part of the cell contains the instruction for life? Golgi apparatus, cell wall, nucleus, lysosome, nucleolus. This should be nucleus. The smooth and rough endoplasmic reticulum both synthesize a molecule. The smooth produces dash and the rough produces, separate the blanks with comma then space. Smooth produces fat and rough endoplasmic reticulum produces protein. Which organelle is necessary for cell division? Cell division, that is chromosome. Chromosome. Choose a symbol on the keyboard that best corresponds to DNA. Choose a symbol on the keyboard that corresponds to RNA. So I think this will be this way. The central dogma of biology is a synonym for the flow of information in a cell. What is it? DNA, RNA to PNA, RNA to protein, protein to DNA to RNA, RNA to DNA to protein, or DNA, RNA to protein. Centered on biology, that is DNA to RNA, then protein. This should be the correct answer. In eukaryote, DNA is scattered throughout the cell. So is it true or false? So in eukaryote, DNA is don't scattered. So this is false. In DNA, A pairs, adenine pairs with guanine, cytosine, thymine, uracil. Should be thymine. And what is the effect of a mutation that does not change the amino acid that the codon codes? Nothing. Truncated works WL, works quadruply well, does not work at all. So this should be nothing because that does not change the amino acid that the codon codes for. Next, the two main type of secondary structure are alpha spirals, helices, fingers, helices, uh, and beta seeds, fingers, seeds, spirals or seeds. So actually alpha is a helix, beta is a seed. So I think this should be the correct answer, alpha helices and beta seeds. So these are the question. So now I will submit it. Okay. So I will see the score of this practice question. So this is 8 out of 10. This is nucleus, fats, proteins, lipid proteins. Correct answer. So this is correct. Why not S is there? Centrosome. Okay. So which organ necessary for cell? This should be centrosome, centriole. That is correct. Correct. Okay. So I can copy paste the which organelles are necessary for cell division. Hmm. Centrioles are mostly present in animal cells and flagellated organisms and structures like spore, gametes, etc., but are present in the plants. These are submicroscopic, microtubular, subcylindrical structures which usually occur in the form of two granules. They are also known as the diplosomes and are present inside a specialized cytoplasm called the centrosphere or kinoplasm. The cell organelle that is responsible for initiating cell division is centriole. With the production of spindle fibers that are an important part of cell division, they are involved in cell division. So it is involved in cytokinesis, where the cytoplasm divides to form two daughter cells. The centriole is a cylindrical structure that is mainly made up of a protein called tubulin. The formation of the centrosome is formed when a pair of centrioles is surrounded by the dense mass of the material called pericentriolar material. Its main function includes the formation of cilia during interphase and the formation of spindle fiber for cell division. The correct answer is centriole. The centrioles are responsible for the formation of basal bodies. cilia, and flagella. They also play an important role in cell division by forming astral spindle poles. So the complex formed by centriole and centrosphere and was termed centrosome by Theodore Boveri in 1888. Okay, so this is actually correct answer. Okay, so I will move to the next part. Next part is this is done. So next part is to again test with biology B. So again I will set this will for again 20 minutes. So okay, so I will start it. After break I will start it. So that is okay. So this is the post test version B. Okay, this is again cell structure. So use this diagram to answer the following 6 questions. And it is labeled 1 to 13. First question is that level 6 represents an organelle that produces sugar. What is this? Level 6 means this portion. Mitochondria, Golgi bodies, chloroplast, vacuole that produces sugar molecule. Okay. So mitochondria produces energy. Golgi body produces protein. Chloroplast produces little parts of sugar vacuole. This stores the water and nutrients. So I would go for chloroplast. Level 4 represents what? Level 4 means this long part. So what is this? Central vacuole or vesicle? Central vacuole for holding nutrients or maintaining turbidity. Actually, role of vacuole is to maintain the turgidity. So I will go for it. Central vacuole. So the level 10 is the internal layer of a substance that surrounds the cell. This is called the cell level 10. Level 10 means this is level 10. So level 10 is the internal layer of a substance that surrounds the cell. This is called cell membrane. The main function of smooth endoplasmic reticulum smooth environment synthesizes lipid. So this should be 2 or 9. So what is smooth? 2, 2, 2. This is rough. 9, this is small. Okay, so this will be 9 and lipid synthesis. Now level 5 present in animal cell, plant cells, some prokaryotes and some eukaryotes, prokaryotes. Level 5, level 5. So this I think in prokaryotes. The organelle labeled will be in higher number in muscle cell compared to fat cell. 1, 5, 8, 10. 1, 5, 8, 8, 10. So I think one is this mitochondria which is remains in muscle cell than fat cell. Okay, so I'll go for 1. Okay, so move for the next part. The backbone of polymer of nucleotides contain the ribose sugar. What is this polymer? Backbone of a polymer of nucleotides contains ribose sugar. So ribose sugar remains in RNA. So what is this polymer? DNA, polypeptide, lipid or RNA? DNA monomer is called nucleotide and is made up of 3 parts: nitrogenous base, ribose sugar and a sulfate group. Which part is correct? So I think no, nitrogenous part is the correct. Nitrogenous base, because ribose sugar remains in RNA and this is not sulfate group, this will be phosphate group. Why is RNA not the main carrier of genetic material? RNA is single-stranded, so it carries half the information. RNA is less stable than DNA. Uracil is toxic and in high amounts. It is lethal. RNA cannot form long chains. So I'll go for RNA is less stable than DNA. What is the justification? So I can write that RNA contains one extra wedge group which makes them less stable and more reactive than DNA. So this is the justification. So move to the next part. Use this diagram to answer the following 7 questions. A delta epsilon T. So this is a single stranded structure. According to the strandedness of this molecule, what type of nucleic acid is it? As single stranded, so I can go for RNA, DNA, RNA, DNTP, NTP. So I will go for RNA. According to the types of bases in the molecule, what type of nucleic acid is it? DNA, RNA, DNTP, NTP. So if there is adenine and thymine bases, so it should be DNA. What is the paradoxical about this image? Paradoxical, it means, so here in this image adenine and thymine. Thymine bases are there, so it must be DNA. Next question is according to your answer to question 11. What must be the final part of the molecule? Phosphate, nitrogenous base, and amino acid, basic group, ribose sugar, or deoxyribose sugar. So 11 answer is RNA. So it should be ribose sugar. According to your answer to question 12, what must be the final part of the molecule? Phosphate, nitrogenous base, and amino acids, a basic group, ribose sugar or deoxyribose sugar. So answer 12 is DNA. So DNA contains deoxyribose sugar. So it should be D. Next, use the following information along with the previous to answer the following two questions. After testing, this nucleic acid is found to be entirety of a thriving and successful alliance genome. Chargaff's rules still apply. A deoxyribose sugar is found in its backbone. What are delta and epsilon most likely to be? As it remains A and T respectively, both adenine, uracil and cytosine respectively, both C. So as in the question it remains deoxyribose sugar, it must be adenine and thymine. What conclusion can be drawn from your finding? Uh, a genome that is comprised of single-stranded DNA is as sufficient as double-stranded. An organism's genome needs C and G to reproduce. The type of sugar does not matter. This must be an exception to Chargaff rule. So I'll go for genome that is comprised of single-stranded DNA. So I'll move for next part. Reverse transcription is the inverse process to the ordinary central dogma of biology. It can be represented as dash to dash. Separate your answer by comma then space. See, inverse process central dogma means DNA to RNA then protein. So it should be inverse to RNA to DNA, RNA comma to DNA. The inverse of the process described in the previous question is inverse process, that is translation and transcription. Inverse of the process described in the previous question. RNA synthesis, ribonucleogenesis, transcription, DNA clip. So this should be transcription. Which statement about proteins is incorrect? A straight chain of amino acids is indicative of primary structure. Beta spiral is indicative of secondary structure. Protein binding with another is indicative of tertiary structure. Quaternary structure is completely impossible. So first per straight chain amino acid is indicative primary structure. Beta spiral is indicative secondary structure. That is correct. Protein binding, so I'll go for beta spiral. So use this diagram to answer the following three questions. So again, some bases are there. DNA template strand contains this sequence 3' GTA, TTG, AAA, GCT, GTG, 5'. What is the complementary strand of mRNA? So 3', then it should be M', 5'. G means cytosine, then T means adenine, then A means uracil. Okay, T means adenine, U, C, then TTT, U, adenine, thymine. So it should be 5 prime CAU AAC and EEU CGS CSC and 3 prime, because last part is GTG. G means C A C, so this is CSC 3 prime. What is the most likely reason that the sequence in the question has been written reverse order? It is customary to do so. It is not customary to do so, but it is easier to understand it in this way. The template strand is read 3 prime to 5 prime, and the mRNA synthesized 5 prime to 3 prime. It should not have been written in this way. So I think this should be the case. The template strand is read 3 prime to 5 prime, and the mRNA is synthesized 5 prime to 3 prime. What protein sequence does this mRNA code for? So it is should be CAU is there, CAU, 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 yes, histidine. Next it is AAC, 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 as ASN. Next part is EAUU, PAG. CGS, CSC, CGS, 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 arginine. Next part is CSC, 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 histidine. So I'll move to the next part. Point mutation has occurred, but the polypeptide is the same. What is this mutation called? Silent nonsense missense indel. Point mutation has occurred, but the polypeptide is same, so it should be silent. And point mutation occurs in a separate polypeptide. The second one interacts with the first one in an epistatic manner, and that causes the original polypeptide to become truncated. What type of mutation is the second mutation? Silent, nonsense, missense, indel. So I'll go for one separate polypeptide. The second one interacts with the first one in epistatic manner and causes the original polypeptide to become truncated. I'll go for nonsense. So I'll check my score. 21 out of 25. So these are correct. Level 5 is prokaryotes. Some prokaryotes, okay, some prokaryotes and some eukaryotes. These are correct. And as one more, this is written same answer. Why is given incorrect? I don't know. Single nature indicates certain okay. Protein. So this should be the straight chain amino acid is indicate primary. Okay, okay, okay. All are correct. No. Okay. So I have completed my test and the score is 21 out of 25. This is not bad. So next part is that okay. Okay, so I will stop here. Thank you.